So this is the fourth part of chapter 3. There's 5 parts. So this is the fourth part, and our focus on this part is going to be proteins. And so these are our third class of macromolecules. They are large, complex, again, macromolecules, organic, carbon-based. They are the most abundant organic molecules in living organisms, so they make up the bulk of our dry mass. Okay, very diverse range of functions, and I'm going to show you examples of these. So proteins can be structural proteins. They can be involved in regulation. They can be involved in lots of different things. So structure, function, and regulation of body cells, tissues, and organs. And so again, we'll talk about some of the different classes of proteins. They're made of amino acids, and so again, proteins are polymers. Their monomeric subunits are called amino acids, and amino acids get arranged in a linear sequence according to our DNA, which we'll talk about towards the end. That linear sequence, again, is going to be according to a gene, and it ends up becoming folded, and so on and so forth. So it is a polymer made up of amino acids. And so we're going to start by going over some of the different functions of proteins. And so some proteins are enzymes. That's a major role that proteins play in our bodies. Enzymes, we're going to spend a whole chapter talking about enzymes, but just to give you a little bit of information, enzymes are involved in chemical reactions. We say that they facilitate chemical reactions. Every chemical reaction that is taking place in your body is going to be catalyzed by an enzyme. Okay. Another word for an enzyme is a catalyst. They are biological catalysts. Their job in a chemical reaction is to speed up the reaction, to increase the reaction rate. And we say that enzymes do this without being changed or consumed in the process. So you can reuse your enzymes. They're very specific for their substrate. So you've probably heard of lock and key, and that's shown here in this image. So in purple is the enzyme, and in green here is its substrate. And so the substrate is whatever the enzyme binds to. And the substrate is binding here to the enzyme, forming something called the enzyme substrate complex. And the enzyme's active site, which is where the substrate binds, is going to be specific. So this enzyme pictured right here is, I don't know, this is a disaccharide. So let's say that this is sucrase. And its substrate is sucrose. So let's say this is sucrose here. Only sucrose can bind. No other disaccharide or nothing else really should be able to bind to that active site. So we say enzymes are specific for their substrate. In this case, the enzyme is catalyzing the breakdown of whatever disaccharide this is. And so here we say yields and the end products have come apart. So this is a hydrolysis reaction. We can also call it a catabolic reaction. So when we break stuff down, that is a catabolic reaction. If the enzyme was facilitating the building up of a larger molecule, we would call that an anabolic reaction. And so enzymes are involved in both catabolic and anabolic reactions. I give you a little list of examples of enzymes. So amylase is an enzyme that breaks down starch. Lipase is an enzyme that breaks down lipids. Pepsin and trypsin are involved in the breakdown of proteins. And lactase is an enzyme that breaks down lactose, right? So, which is a disaccharide found in dairy products. Okay, all examples of enzymes. The second potential function of some proteins will be hormones. Okay, so hormones are chemical messengers, chemical signaling molecules, and they're involved in regulation and maintenance of homeostasis. So in this example here on the right, I have a blood vessel here. So let's say you just ate a meal and your stomach is going to break down the food that you just ate, and glucose monomers will end up in your bloodstream. And so after a meal, your blood sugar is going to be relatively high. What will happen next is your pancreas is going to release insulin. So shown here is insulin, and insulin is a hormone, a protein hormone. And that insulin is going to trigger your cells to take up glucose, to get it out of your bloodstream and to take it up. And so what happens after insulin is released and your cells do their job is your blood glucose levels go back to normal. Okay, so again, hormones are involved in regulating processes and keeping you in a homeostatic balanced state. Okay, so the next function of some proteins is transport. And so these type of proteins transport stuff. They might transport stuff, from one part of your body to another, or from the inside of a cell to the outside of a cell. And so I got two different examples here. So first of all, if we were to take a peek inside of your red blood cells, what we would find is hemoglobin protein. And this is actually made up of 4 separate polypeptides that come together to form a protein. And it's iron containing. So you can see these little dots here are iron. And what hemoglobin does is it's going to transport oxygen, right, that you take in through your lungs. And it's going to transport that oxygen to all the cells of your body because your cells need oxygen for cellular respiration. So that's an example of a transport protein. If we also look at the cellular level, remember this drawing, this image, this is the phospholipid bilayer, right? The membrane. This is the inside of the cell, the cytoplasm, and this is the outside of the cell. Both are aqueous environments. And so sitting in this plasma membrane is this big purple globular structure. This is a membrane protein. And what membrane proteins will do is they will allow molecules to pass from one side of the membrane to another. And that might be through diffusion, so it's the passive transport without the requirement of energy, or protein transporters can also be involved in active transport, which is the pumping of materials across a membrane against a concentration gradient. And that requires energy. We're going to talk about those two processes when we get to chapter 5, I believe. But for now, I just need you to know that there are proteins in your membrane that help to move stuff across that membrane. Okay, what else? We have some proteins involved in structural support. Some examples here, your hair has keratin protein. We also find keratin in horns of horned animals and feathers. We find silk fibers, which are protein in nature. We find them being produced by insects and spiders. Insects would build cocoons and spiders would build webs with that protein fiber. And then also inside of us and other animals, we have collagen and elastin proteins. And so in this figure right here, I'm showing you a type of tissue. It's connective tissue called areolar connective tissue. And we've pulled out one of these thick fibers. And if we look at it, it looks something like this. This is collagen. Okay, so again, it's protein, it's proteinaceous. And it's kind of like a rope, a couple of ropes that have actually been kind of wound around each other. Provides a lot of structural support. So we got collagen and elastin proteins that are going to be found again in connective tissue in animals, like skin, tendons, and in your bones. Okay, proteins can also be found in defense. These are my favorite proteins. So defense, we're talking about antibodies. And so you have specialized immune cells called B cells that are going to be responsible for producing antibodies to protect us against disease. That could be from viruses or bacteria or any other pathogen, anything really foreign. And so what antibodies do is they will recognize stuff, bacteria, viruses, as being foreign, not belonging to you. And those antibodies will attach and then neutralize those harmful substances. Okay, we're getting close here. We got 2 more left to go for different types of proteins. Some proteins are involved in movement, so we would call these contractile and motor proteins. So in this example here, I'm showing you a muscle fiber, which we'll actually take a look at in the lab. If I were to look inside this muscle fibers have to be able to contract and then expand. That's how you move things, right? That's how I'm moving my arm right now, is contraction and expansion of muscle fibers. And so how is that done? So if we were to look inside of the cell, this is what we would find. We would find actin and myosin proteins, and their movement is going to allow this whole cell here, or fiber I should say, to contract and expand. We also find motor proteins inside of cilia and flagella. We'll look at these two structures in chapter 4. These are both involved in movement of cells. Okay. The next and last type of function that proteins might have is they may be involved in storage. So storage of amino acids. And so, for example, you may have heard of casein. Casein is a milk protein, which is a major source of amino acids for baby mammals. Plants, if you take a peek inside of their seeds, you would find storage proteins in there. And then lastly, in the picture here, we have eggs, bird eggs, okay? And if I were to take a peek in here at the egg white, we would find a protein called ovalbumin. And ovalbumin is made of amino acids. This can be broken down, and these amino acids can be a source for the developing embryo inside of that egg. Okay, so storage of amino acids. Okay, so let's start to talk about what proteins are made of and how they're formed. There are many different types of proteins, right? We just saw a bunch of different functions. Proteins are going to be different in function, they'll be different in size, they'll be different in molecular weight, and probably most importantly, proteins will differ in shape. And so shape is critical to function. Remember lock and key when we looked at enzymes? A protein has to fold up properly and be in its proper three-dimensional shape or conformation in order to work properly. Some things that might affect protein shape. So if we were to change temperature or pH, proteins have a very specific narrow window of temperature and pH where they're going to function correctly at. If you get outside of that narrow range, they won't work. And we'll talk about denaturation. I'll introduce it here and then it's brought up again at the end of this section. But denaturation means to lose shape and therefore lose function. So again, outside of temperature or pH range or exposure to some kind of a chemical may cause that to happen. Proteins are made up of a set of 20 amino acids. Okay, so there are 20 naturally occurring amino acids. Some amino acids your bodies can make, and so we call those non-essential amino acids. You still need them, but your body can make them. Essential amino acids are going to be those amino acids that your body cannot synthesize, so instead you have to get them from your diet. Okay, so there's 20 total, and I believe there's 11 essential. All 20 amino acids have the same backbone, and so that backbone is actually here for us. And you should learn this, okay? I'm going to ask you to sketch it in lecture, and then you're also going to be required to build some of these in lab, and so you should be able to sketch it. So this is the common backbone, okay? So every amino acid is going to have a central carbon, which we can call either the alpha carbon or some people call it the asymmetric carbon. Either is fine. That carbon will be covalently attached to a carboxyl group on one side and an amino group on the other side. It will then be bonded to a hydrogen. And then up here, this R, this is the side chain. This is what makes the 20 different amino acids different. That R group could be a hydrogen. It could be a CH3 group, or it could be a larger group. And I'm going to show you a picture of the 20 different amino acids on the next slide. And again, I want you to realize that the only thing that's different is what's here. Okay. I also want you to realize that when amino acids are in aqueous solution, they will ionize. And so on the next slide, when I show you the 20 different amino acids, you're going to see some charge. And so I need you to realize that this carboxyl group will give up this proton. And so this becomes a negative. And this amino group will accept a proton. And so this becomes NH3, and it's a positive charge on the end. Okay, I don't want you to get hung up on that. I just want you to realize that that's what happens. Okay, so here are the 20 different amino acids. You do not have to memorize them. Okay, I don't need you to know their shape, really, or their structure. If I'm going to ask you a question about amino acids, you will have access to this table. Okay, what I do want you to realize is the different amino acids differ in structure, right? But they also differ, that R group gives it specific properties. So every amino acid here in this top group, so we got glycine, alanine, valine, isoleucine, methionine, and leucine. Their R groups, so what's highlighted in blue here, they're all non-polar, okay, hydrophobic. So these amino acids are going to tend to be towards the center of the protein as it folds because it wants to be away from water. Very different from what's down here. So serine, threonine, cytosine, glutamine, asparagine, and proline all have polar, uncharged R groups. So they're polar, so there's some polarity here, so they will associate with water. When I come over here, histidine, arginine, and lysine, if we look at their R groups, their side chains, they're positively charged, so NH positive, NH2 positive, NH3 positive. And so these positively charged R groups are going to be hydrophilic, want to associate with water. We then have negatively charged, same thing. These are also going to be hydrophilic, okay, aspartate and glutamate. And lastly, these are some non-polar, we call them aromatic because they have ring structures. So phenylalanine, tyrosine, and tryptophan. These are all going to be hydrophobic, non-polar. So you got to remember what polar versus unpolar means, what it means to be charged, and then realize that non-polar groups are going to be hydrophobic, where polar and charged groups are going to be hydrophilic. Okay, so that's going to really dictate how the protein folds. So think about form equals function. Which amino acids are present, that's going to dictate how the protein folds. Okay. So we saw the 20 different amino acids. We know that they all have the same backbone. It's their R groups that are different. And so how do we put amino acids together to form a polypeptide? So in this figure here, I'm showing you dehydration synthesis. So here is one amino acid, and here is the second amino acid, and we're going to put them together through dehydration synthesis. So what's going to happen is the hydroxyl group from amino acid number 1 is going to come off, and so is this hydrogen here from amino acid number 2. So we're going to pull out water, H2O, and we're going to connect, right, covalently bond this carbon to this nitrogen. So that's shown here. This is now a dipeptide made up of 2 amino acids, and the bond right here holding these together gets a special name. It's called a peptide bond. So remember, the name of the bond between 2 sugars in carbohydrates is going to be called a glycosidic bond or a glycosidic linkage. In lipids, we called it an ester linkage. And here we're calling it a peptide bond. It's just a covalent bond, but we give it a special name. Okay, and again, this is a dipeptide, just like we saw disaccharide, okay, made up of 2 monomeric subunits. And again, it's put together by dehydration synthesis. And I just wanted to show you we can keep adding, right? And so we've got 1 amino acid, 2 amino acids, and now we're about to add the third. Okay, this third amino acid gets added just like the second one did. Water comes out, dehydration synthesis, here's that new bond. Okay, so again, the backbone, which is shown here in purple, is the same. All 3 amino acids have the same backbone. The side chains are what's different. Okay, these 3 side chains all happen to be hydrophobic, but they're going to be different. Okay. A complete protein is going to consist of 1 or more polypeptide chains with hundreds of amino acids all joined together in a very specific linear order. Again, you can think of the 20 different amino acids. You can think of them like letters of the alphabet. They get put together very specifically to make words, okay, according to your DNA. So there is really an almost infinite number of combinations of these 20 letters, these 20 different amino acids. And so you can get a variety of different proteins, right? Almost an infinite variety. What your protein looks like and what it does is coded for by your DNA. And I'm going to show you an image at the end of this showing you transcription and translation. So just as a precursor, going from DNA to RNA that's called transcription. And then going from RNA to protein, that's called translation. So again, at the end of the chapter, I'll talk about both of those again. Just wanted to introduce them to you here. So the proteins that you have in you is determined by your genes. Okay, and I want to show you shape. So again, how a protein folds up, and a complete protein can be one polypeptide chain fold up or multiple, right, interacting together. That's going to tell us what the protein does. Okay, we call a protein shape, it's its three-dimensional shape, or we can call it a conformation. That means shape, same thing. So what a protein does, what its function is determined by its shape. It's determined by its conformation. Okay, and its conformation is determined by the sequence of amino acids, which is determined by the gene. Okay. Here's an example. We've got over here on the left, this is an antibody protein. And here on the right is actually a protein from the flu virus. And you can see how these two proteins match up perfectly. So your antibody is going to have this really specific site called the antigen recognition site, and it's going to bind, lock and key, just like enzymes do, perfectly to this flu virus. Okay? Okay. And that's true, again, of enzymes, of protein hormones, of defense proteins. Okay? Form is going to equal function. 3D shape is important. So how does a protein get its 3D shape? So again, your DNA, right, to your RNA and now to your protein, but how exactly does a protein, fold? What does it look like? What's the series of steps? So there's 4 levels of protein structure, and the names are really easy. Primary, secondary, tertiary, and quaternary. Okay, so 4 levels of protein structure. Here's the first level. So again, this is called the primary structure. So what do we have here? We just have a string of amino acids, right? Glycine, proline, threonine, glycine, threonine, glycine, so on and so forth, all the way. So we got like 25 or 27 amino acids all in a string here. Okay, this is the primary structure. So just to give you a little bit more information, again, you have your DNA. A copy of the gene is made. That's called RNA. The RNA goes to the cytoplasm. And in the cytoplasm, the RNA is read by a ribosome. And the ribosome actually will put these together based on what the code is in your RNA, which came from your DNA. And so the ribosome is going to literally take these amino acids and put them together in this string, right, through dehydration synthesis, according to your gene. And that's the primary structure. So it's literally the string of amino acids. What happens next? The protein starts to fold. So secondary structure. So within the backbone, there's going to be hydrogen bonding that's going to occur. And that hydrogen bonding is going to lead to 2 structures. Beta pleated sheets, which look like this, and alpha helices, these coiled-like structures. So again, these are associations between the backbone of the amino acids. That's going to give you secondary structure. What comes next is tertiary structure. And so this is going to result from interactions between the side chains, okay, of those amino acids. The tertiary structure is the 3D shape of the polypeptide, okay? What kind of interactions? Hydrophobic interactions, okay? So this is an example of a hydrophobic interaction. Both of these side chains are hydrophobic, and so they're going to kind of tuck themselves together in the inside of the molecule, in the inside of the polypeptide. Ionic bonding, and so that's shown here. You've got charged regions. They're going to be attracted to each other. You also can get hydrogen bonding. So polar regions attracted to each other. And then these are called disulfide linkages. So sulfur, if the side chain contains sulfur, they can actually covalently bond. So that's the tertiary structure. The the polypeptide is completely folded. This is its 3-dimensional shape. Now, if a protein is made of multiple polypeptides, then it will have a quaternary structure, that 4th level. We would consider this level the complete protein. Okay, so again, this is going to be 2 or more polypeptide chains that are associated with each other. And many proteins are made up of multiple polypeptide chains. Okay? Again, the overall structure of the protein determines its function. And so on this slide, there's just a review of the definitions of each of those structures. So again, we go from primary, string of amino acids, secondary, beta-pleated sheets and alpha helices. These are interactions of the backbone, between the backbone amino acids. Tertiary structure, this is going to be a fully folded polypeptide. And again, this is interactions between the side chains. And then lastly, if you have more than one polypeptide chain to a protein, you end up with this quaternary structure. Shape equals function. If a protein doesn't fold right, it ain't going to work. Okay? So what if a protein doesn't fold right? So if a protein is subjected to high heat or changes in pH, or it's targeted with certain chemicals, the protein structure can become disordered. Polypeptide chains could unfold. We call that denaturation. We say the protein has become denatured. And that happens, okay? As your proteins are being produced, so as amino acids are being put together, we get primary structure. As the protein starts to fold, so secondary to tertiary, there are actually going to be helper proteins that are sitting there waiting in the cytoplasm, ready to help proteins fold. We call those molecular chaperones. So that's right here. So molecular chaperones are going to be proteins that are going to mediate the folding of other proteins. They help make the folding process more efficient, okay, and they help to make sure that proteins get folded properly, okay? The last thing I wanted to mention quickly is what about if there's a mutation? And we're going to talk about mutations when we get into the chapters on DNA, but I just want to let you know that a mutation is a change in your DNA sequence, and that can show up in protein shape. Some mutations are silent, they don't do anything, and some mutations have drastic effects. I'm going to show you a mutation that has drastic effects. So you've probably all heard of sickle cell anemia, okay? I'll take you back to your red blood cells. I showed you this already, but your red blood cells have a protein called hemoglobin. And hemoglobin, it's made up of four polypeptides. So this is the quaternary structure. Its job is to carry oxygen to all the cells of all the tissues in your body. People with sickle cell anemia have a single amino acid substitution. They have a single change in one amino acid that affects the beta hemoglobin protein. So let me show you this. Okay. So this is the normal sequence of amino acids in the beta subunit of hemoglobin. People with sickle cell disease at position 6, instead of having glutamine, they have valine. Okay, now again, a single amino acid substitution may have no effect. In this case, it's drastic. So what happens? So this is how the beta subunit normally folds. This is the secondary and tertiary folding of it. This is what the beta subunit in somebody with sickle cell, this is what it looks like. Very different. So again, shape equals function. So it doesn't fold properly. And so this is what normal hemoglobin looks like. This is the quaternary structure. And this is what sickle cell hemoglobin looks like. So normally, these hemoglobin quaternary proteins, they stay separated from each other. They carry their oxygen. Red blood cells have a nice concave shape. Everything's working properly. People with sickle cell, whose hemoglobin looks like this because of the beta hemoglobin protein, their hemoglobin doesn't stay nice and separated. Instead, it forms these crystal fiber-like structures, which greatly decreases the capacity of these molecules to carry oxygen. And it also changes the shape of the red blood cells. And so they call this the sickle shape, or we can call it like a crescent. This is a big problem, right? So number one, you're not going to be able to get oxygen to your cells because your beta hemoglobin can't carry it. And secondly, because your red blood cells are misshaped, they're actually going to get stuck in capillary beds. And so people with sickle cell anemia, when the red blood cells get caught like that, that can damage organs. It can cause excruciating pain, lots and lots of problems. Okay, so again, form equals function. If a protein doesn't go through its primary, secondary, tertiary, and quaternary folding properly, it ain't going to work. Okay, a change in your DNA could equal a change in amino acids, which would cause a change in the protein. We'll talk about sickle cell disease again later in the semester, but just so that you know, a very new therapy, cure, I'd like to say, for sickle cell has been developed, and it was actually, it just made it out of drug trials just this past, as a couple months ago now. There's a technology called CRISPR technology where they actually have edited the gene, and they've been able to fix it, and the results are extremely promising. So, form equals function. That's the end of this part of the chapter.